We're going to be talking about some new features in the DNASTAR Lasergene software suite that pertain to auto-annotation of sequences, and we'll focus mostly on plasmid sequences today. But first, I'd like to give you just a brief background as to what our company offers in terms of software. We have software in three areas: molecular biology, structural biology, and genomics. And they're integrated solutions so that we can do work in one area and transfer sequences and analysis from one area to the next. And today we're really going to focus on some of our molecular biology tools. And the molecular biology suite will have things like Seq Builder for cloning and for sequence editing and annotation. But we also have these other software suites, genomic suites, that offer specific tools and abilities to work with NGS data and both assemble and analyze. So there's some different views of aligned data, SNP reports, different kinds of analysis. So we have a number of webinars that cover genomics, and those are some topics that come up from time to time. We also have structural biology tools. Structural biology will offer some new software that allows us to do things like protein modeling and protein-protein interactions and antibody design. So today we're going to be focusing on that molecular biology suite, which is one of the three areas. And the molecular biology suite has really been really is expansive in the number of different things that it can cover. And that includes things like phylogenetic alignments, multiple sequence alignments, primer design, different ways to edit and annotate sequences. So it's been a suite of tools that we've had for over 30 years. There's wide-ranging function, and recently we focused on some expanded functionality for annotation. And really today's webinar is going to focus on auto-annotation, and this is all done in our Seq Builder application. So Seq Builder has many different functions. Some new functionality was added recently in terms of cloning. So we have some videos and materials that support different types of cloning methods, including a new cloning wizard. Seq Builder also has primer design and the ability to create linear and circular maps. And really today what we're going to look at is some auto-annotation, and that's to take unannotated sequences or annotated sequences and apply annotations that are curated in a database. And so that will really be the focal point today. So automated plasmid annotation, it can be as simple as a single plasmid, or it could be a large plasmid with hundreds of features, or it could be a collection of plasmids, dozens or even hundreds of sequences, and it's really meant as a time saver in that we can automate the process, which is a very manual. Otherwise, it would be a very manual process of adding annotations one at a time to these sequences. This can streamline and make what's a very kind of a tedious job very quick. So there's multiple different aspects to our auto-annotation tools. The first of these is really the database that's used, and we had an expert annotator go through plasmid vectors and create a subset of features that are curated. So it's very important to go through all the potential plasmid annotations and curate them so that they aren't overly duplicated, that they're accurate. There's many annotations from commercial vendors that are inaccurate. They're just simply not the right annotations. We have to go through those and make sure that each of those annotations is accurate. Pull out the ones that aren't that aren't good enough, and have a collection then of mostly unique and very accurate annotations. And then using that database, then we can scan against it using our own algorithms to match the query sequence, the unannotated plasmid, to this database. So we have our own algorithms that we also use in our genomics and our NGS that's very powerful, that can take large sets of sequences and compare them and find the matches. So we have a great capacity then due to this powerful algorithm that is powering the whole thing. And so that gives us the ability to annotate single sequences or large batches of sequences, large and small plasmids. This tool is initially intended for, sequences, up to about a few hundred KB. You might be able to stretch that a little bit larger, but it's mostly plasmids and some of the larger environmental plasmids that you might see. And then there's a UI. So we have a user interface that allows you to interrogate and look at the annotations. So the workflow can be fully automated where you just accept all the annotations, or you can look at them and pick the ones that you want to add to your sequence. And that's a very, very important piece of functionality so that you can further confirm and make sure that the annotations that are being added aren't already there on your sequence or are the type of annotations that you want to add. And so this database then can also be expanded. It's possible to take our curated database and then add additional features, harvest features from new plasmids or from different types of sequences and have your own collection of features, and then the ability to look at that and curate that database further. So it gives us a lot of flexibility, and it gives the user a lot of control to make sure that whatever annotations they're adding are the most accurate. So that's what we'd like to do today. With that, I will jump out of the PowerPoint and we'll go into some software so I can show you what the workflow looks like. So we're going to start here by launching Seq Builder, version 14. So this is actually 14.1. And I'm just going to open a sequence here that's unannotated. So we'll open up a PBR322 FASTA file. And so we have a circular map. There's no annotations, just a few enzymes applied. So this would be the most basic application here, would be to have just an unannotated plasmid, and we go to the feature menu and click annotate sequence, and it gives me an interface now for some parameters. So what's driving all of this is a very powerful alignment algorithm, and we give the user access to some controls to say, I either want perfect matches only, so that's 100% match, the feature can't have any mismatch at all, or I can use the default settings, which allows some mismatch. So, 98% coverage and a match percentage of 95. So that allows some wiggle room if you're trying to pull in features that may have a couple of base pairs different from the sequence that you're from the plasmid sequence. So you have the option to run different stringencies then of that matching. So whatever you pick, you can select OK, and now the algorithm is running. It's searching that plasmid sequence up against the database, and we get a results window then. And so the results window, I'm going to just minimize a little bit so we can see both, has in the upper left-hand corner, I can accept checked. And what that means is that in the table in the bottom, you can see that we've got 6 features that are reported, and because we had an unannotated sequence, all 6 of these are unique, right? They aren't already on the sequence, and so all of them are checked by default, meaning that if I click this button, all 6 of these annotations will get added. And now I can go in and look at the annotations. So I have this matched feature that tells me the top annotation is the TET-R promoter, and I can see how long it is. The match quality is actually a histogram. It's a perfect match, so everything is just green. If there's mismatches or gaps, you'll see a difference. A mismatch will be a yellow base, and a gap will be represented by a dash. And then I have this matched feature area. So again, that's the TET promoter and pre-existing features. So if I had annotations on this sequence already, they would show up in the neighborhood, or in some cases, we've already got that feature. And I'll show you examples with some other sequences where that's used. And again, the same with this blue and yellow. This is really allows me to do things like replace features that are there or add features. And it's telling me if there's duplicates that are found. So what you don't want to do when you're auto-annotating is add a bunch of duplicate features. So if you've got some features there, I don't want to add duplicates. If my database has some features that are very similar, like amp resistance genes, for instance, there may be several different versions of that gene in your database. I don't want to add multiple copies of the same thing. And so we use this interface then to make sure that whatever annotations are being added, they're unique. So I can look at each annotation now. I'm clicking through the table and see what they look like. And I get the query sequence, which was my PBR322 gene. So I'm looking in the table now. I get the type, so I get a feature type, and I get what the matched feature is called, and then I get the alignment scores, the position, then which strand it's on, the length, and then a description of that feature. So I get some of the GenBank information coming through in the table. So I can look at this and say, for example, that I want to add those annotations. So I can accept the checked, and now my plasmid sequence, you can see I've got annotations that appear on it. So by accepting the checked, I get those annotations added, and I also get a little history here that just tells me, which features were added. Now, of course, SeqBuilder has wide-ranging functionality, so you can, if I want to change how things look, there's multiple different ways to do that. I can do it on a per-sequence basis. So if I want to reposition, a feature, I can double-click on it and move it. And if I want to, so here's a smaller feature. If I want to be able to read the label, I can right-click and detach the label on it, and then I can zoom into an area. So it's got a high degree of flexibility as to how these features can look. If I want to change the style that's a default, I can double-click on a feature, right, edit a feature style. So I can make the color, maybe I want to make the color a little bit different. So there's some flexibility there as well to make the features look different, to reposition them. So I can put them on the backbone or inside the backbone. And then there's ways to set these features. So these are new features that were, and I'll show you that in a little bit later in the webinar. So I can set new defaults for these features that are added as well. So if I always want my CDSs to be a light green, there's a way to change that so all those features stay light green as well. And I'll show you what that interface looks like. So that's a single small sequence, relatively small. Let's close that and try another sequence here. So this will be another unannotated sequence called superplasmid, which is just a large plasmid that has lots of annotations. So again, we'll just do an annotate sequence here. And we can see now that the results are returned. And now there's 86 matching. 73 of those are unique. So if we read this print up here. So what that means is that we've got 73, which the algorithm says these are unique features. But the database has some duplicates. So if I look, I'm just going to scroll down here through this feature list. And you can see down towards the bottom, there are some that are not checked. And so what that means is that the annotator said we're already going to put a feature here already. So if I sort by position, I'll just show you one of the examples here. So here's an origin. Here's another origin. It's labeled a little bit differently. And now when we look at our match quality, you can see that there's a little bit of mismatch on this one feature. So there are, for some of the common features or elements and vectors, there's slight variations of those in the database. And the detector says, well, there's two of them. One of them is 100% score and 100% coverage. It's a rep origin feature. And it's called F1 origin. It's from LAC-Z alpha. And then there's another version of that has a little bit different name. It's the same length. And it's very close. So if I look at this one, this one's perfect. This one has a mismatch. So there's two very close database features, but we only are going to annotate with one of them. And that's what's very, very important, is to have that control so that I'm not duplicating and adding very, very similar or the same annotations on top of one another in this annotation step. And so we're going to add, so some of these are unchecked that already have, multiple copies of the BLA or the AMPR gene. We're only going to add that one copy. So again, there's 86 features. I can go through and look at them, or I can just say accept just those that are checked. If I see one that I don't want to add, I can ignore and uncheck it, or I can select multiple and uncheck. So I can deselect as well in this left-hand column to say, I don't want to add that particular feature. And now we can add these features to our super plasmid sequence. And there we have the annotations added now to this 30 kb sequence. And now we have many more features, and we can see in the annotation summary and the history of the annotations, I can see all the annotations that were added. So even something that size, very fast, so very fast annotation. And so this can scale up. We can go larger. I've done up to about 300 kb for some environmental plasmids, and that's about what we've designed this particular tool for, is sequences in that size range. Now it's also possible. So these are single sequences. It's also possible to do multiple sequences. So I'm going to open what's called a project file in SeqBuilder. And a project file is really a set of sequences that I can collect. And in this case, I'm going to collect the plasmid vectors, which are the cloning vectors that it's really the set of vectors that we include with our software as part of for our cloning tutorials. We have these, and we have some vectors already, and I converted them to an unannotated format just to make them to go through the steps. So now I have this folder that contains, a couple hundred different plasmid sequences. Now I could select the whole thing. In this case, I'm just going to. It takes about a minute to do the annotation. I don't think we want to watch for a minute, so I'm just going to select a collection of them, just some different ones. Deselected here. I'll just take a block here. This might be like that. That's easier. Okay, so we're just going to select a group out of there. And so now the query is going to be multiple, those multiple sequences. So I'll have this first column that will have multiple sequences in it, and identified by the plasmid name. And it should come through here in a few seconds. And still returning. So we've got, now this brought back 2,375 features, and 1,989 of them are unique. Again, these are unannotated sequences, and so now we have a much larger group. And you can see now the query column, there's the M13MP2 plasmid, and as I scroll down, we can see, I selected a big selection of them. So we have all the different sequences, and again, I could go through, or I could just say, accept checked. And I'm not actually going to click that because it'll take about a minute or so at that many features, 30 seconds. So again, accept checked, and all these annotations will be added to this group of plasmids. So again, a very useful feature, scalable at least, into the hundreds of plasmids, possibly more depending, on your computer resources. You'll want a little bit more powerful computer if you're doing very large sets of sequences. So that's another application, multiple sequences. Now I'm going to show a little bit different functionality. I'm going to open an annotated sequence. So in this case, we'll do a PBR322 that's in a SeqBuilder format. So this is an annotated sequence already. So I have, a circular map here with some features that are on it, and something that's really common with vectors, especially when you get them from a vendor, you download a file, is that those annotations are either incomplete or they're not accurate. And we know that because we've gone through the list that we get from vendors, and as we start to curate them for our cloning functionality, you see that a lot of features are just flat out missing, or they're just in wrong coordinates, and we correct them manually and go through and make sure that they're accurate. Now we have an auto annotation tool. We can apply the auto annotations and improve them in batches. So we can see here we have a number of different features. When I click on them in our map, if I split the window, I can show a feature list, so I can get the information about those features. So here's the blah gene, and so in this lower pane, then I can get all the annotation information by clicking on the feature, double-clicking. There's a signal peptide. So I have some annotations on this sequence. So I'm going to annotate this sequence now. And so now I get 8 features that are matching this particular sequence, and 5 of them are unique. And when I look at them, I can see, well, here's one that would be added, the TET-R promoter. And I can see that on my annotated plasmid, that feature just isn't there. So that's one I might want to add. And then I have another promoter, an AMP-R promoter. Now you can see in this pre-existing features, here's some gene annotations in the neighborhood that start to show up off to the right there. And now here's one that wouldn't be added. So the TET-R, it looks like we've already got that. So the pre-existing features, they're already there. So this new annotation, it's unchecked by default, and I can look at the picture and say, yeah, we've already got that annotation there. I'm not going to add that one. And same thing with the rope gene. Again, AMP-R, and here's another promoter. So I can kind of go through and look at that and see, oh, here's another new feature, and here's another one. So this one's a little bit more interesting. There is a pre-existing gene there, but the new annotation shows that it's a 100% match, that it might actually, or it's a 99.6% match. So I might have a decision here on this particular feature to replace or ignore. And so there is a pre-existing feature. So I can, do the replace or ignore, and by replacing, I can take the new feature and replace that existing feature. So I've got some options on some of these features, again, to improve the annotations that are on an already annotated sequence. So again, I can accept the checked, and now I get some new features that are added that weren't there previously. And again, I can do that in batch. If I've got a whole set of annotated sequences and I want to try to improve the annotations, I can load all of them in, do the exact same thing, and get an improved annotation. So that's how the auto annotation part works on different types of sequences. Now I'm also going to show you what's the database. There is a user interface for that as well. I'll minimize this. So under features, there is a feature library manager. And this manager then allows me to look at what's in that database. Right? So these are the curated features of plasmid parts that we supply with the software. So when you install the software, there's a file that contains this information that will be placed on your computer. And you can see the whole list here. I'm just going to scroll through it. And I can search, if I want to look for a particular one, blah. And when I have a feature, I can visualize the feature in different ways. I can even edit it. If it's one of my user features, I can change it. Here, these curated features are actually locked down, so I can change how it's visualized. So I can say, oh, I don't really need the enzymes, and maybe I want top and bottom strands, and maybe I want translations or not. And then I can also look at the GenBank qualifiers here, so I can see what the qualifiers are. And then there's also stylings. So if I want to change the style for the feature, there's a button up here that allows me to go and edit that style. So again, I can change the line color, fill color, and the style for that feature in this area. So again, there's the whole feature list. Now there also is some capability to add my own features. And so there's a DNASTAR, and this is kind of the archived list, and then there's also a user list. So the user list are features that I've added, and right now I've deselected them. What that means with these checkboxes is that once you deselect, that means when I do an auto annotation step, these features aren't going to be included in the search. So I have a way to say, I don't want to use everything in the user or the DNASTAR database. I might decide that, I really don't want some of these features to be part of the auto annotation, maybe for particular sequences. Then maybe I come back and can have some control then over which sequences are used. And so I can select a group and say, I want to use all of these custom features when I do my auto annotation steps. Now, the way to add sequences here, there's multiple different approaches. I can drag and drop sequences right into this area. I can import features and go and grab a sequence and say, well, let's take all the features off of this PBR322 and bring them in. And so it just strips the features out. So now I have all those features added to my custom database. So that's another way. Another possibility is if I have a sequence open. So I'm going to open up a big sequence. So this is a different kind of a plasmid now. So this will be a environmental plasmid. So it's a big one, 244 kb in size. And so maybe this is something I'm working with, and I decide that, let me get this view here, it's a little bit bigger. I'm just going to set this into, use my Seq Builder windows to show a map and the actual sequence data below. And so if I have something that is full of features like this, I could select the whole sequence or just the range and say, I want all those features. So in this sequence view, then I have that range selected. I'm just going to right-click here and say, send those features to my feature library. And so it just did that. It just harvested all those features, and there they are in that longer list. And so now I have a much larger list of features. And of course, these aren't typical cloning vector features. These are, from an environmental, more of an environmental plasmid. So it's a whole different feature set. I can also make some modification here. So the name that appears, so this is called mock-R. If you look in this bottom, in these GenBank qualifiers, there's one that DNASTAR created and uses called DNASTitle. So I can edit the name here. So if I want the name to appear differently than what is used by default, I can rename that. You can see that was renamed up here. I can also rename here by right-clicking, and it will update the DNASTitle. So I have control over the names, how the features look. I can create a subset of my own custom features, and I also have the DNASTAR highly curated features for plasmid annotation. And with that, that's all I have today. It's a workflow that is going to be very, very useful, a great time saver for those that are looking to annotate sequences, in particular plasmid sequences. With that, I'll take any questions. Thank you.
Hi, Matt. Thanks for that presentation. We have 3 questions that have come in from our participants. And the first question is, can I use the auto annotation tool for sequences other than plasmids?
Yeah, great question. There certainly is capability to import features really from any source. So it could be a linear molecule, a collection of sequences in GenBank or in Seq Builder format or .seq format. It's a robust mechanism to strip features off and add them to this user database. Again, the scope is intended towards not genomic type annotations, but it could be things up to 300 kb in size. So it does have that flexibility built in to be a more general purpose annotator.
Thank you. The next question is, will there be additional new capability in Seq Builder version 15, and if so, what?
Yes. So there is some continued development. So we released this new annotation capability in 14.1. You will see some new improvements as well in Seq Builder in the future. Some of the look and feel and of the different maps and some of the different views in Seq Builder will be updated. So certainly if you're a Seq Builder user, you'll want to check out both 14.1 and any kind of improvements. There should be plenty in the next release.
Okay, and here's the last question that we have time for, although we just had another question come in from a participant. I will have Matt email you an answer to your question after the webinar. So for now, we just have time for one more, and that is, can I create customized sequence views and windows in Seq Builder?
Yeah, there's different mechanisms to change the views, and we do have some other webinars that show how to do that. But using these views on the side, I can go through and create different types of maps. And so these selections on the left control what you see on the right. And I can also create additional splits in the window. So you see I can grab these bars and make this view as flexible as I need it. So I can have, even do a vertical split in a window if I wanted to, depending again on what your need is. I can really rearrange this how I want it. So if I prefer that sort of a arrangement, I can certainly do that. Have a linear map on the top and a with the controllers off to the right. I can also have, you can see there's circular maps, so I can have linear maps and circular maps going at the same time, or sequence views, or feature lists. And then if I save this document, of course, those windows will be preserved, with that save. So it's a very, very adaptable and flexible program.